Hello, I'm Rose Higgins, the CEO of HealthMyne. We're a Madison, Wisconsin-based applied radiomics company which focuses on oncology. Dr. Robert Gillies, a number of years ago, rather famously stated that images are more than pictures, they're data. And that's really the essence of what we're going to spend time talking about today. If you think about the fact that over 90% of all healthcare data comes from medical images, and you think about the fact that hidden within each of those images is a treasure trove of untapped data that really helps describe tumor biology, and that's the nature of radiomics. Our job is to go in, to mine that data from these medical images and present that data back so that you can help to predict which patients might be benefited from particular therapies, to understand which patients are more likely to have positive or negative outcomes, and also to use that data to help personalize the treatment strategies for those individuals. And so that's the nature of the work that we do. We do this work really as we map a person's cancer journey. If you think about the fact that across all of every cancer journey, there is an image that's a hallmark of a particular stage in that journey, we're able to then ascertain from each of those points along this timeline different elements of information that help us understand whether or not something is actually working as expected, or if it's not, allows for the treatment to be modified and adjusted, helps us understand whether or not the response is as expected, and again, if not, to tailor treatment in accordance with that. So think about every single person that you know along your journey who has encountered cancer and what they've had to undertake. Well, part of that is this imaging journey, and we want to be able to take as much benefit from that experience as we can by providing this new information to help inform how to make this the most optimal journey possible. With that, the process requires us to really kind of get in there and take a look at what's going on in this imaging data. So if you look here on the left-hand side of the screen, what you'll see is a CT scan of lungs. And typically, the way in which the read or the report that accompanies that image states tends to be rather qualitative in nature. Something's abnormal, it may be suspicious, there may need to be follow-up. And there may also be measurements that look at the long and short of that particular lesion, but it doesn't go beneath the surface of that. It doesn't tell us what else is going on in that particular lesion that could be useful information. And that's the move from the qualitative to the quantitative. And that's really what we do as it relates to extracting radiomic features. We extract over 1,600 radiomic features from any given lesion. And from there, then, we're able to provide more specific detailed data that can help an individual understand what's really transpiring as you're looking at these two time points that are depicted on the right-hand side of the screen. You'll see that the data is far more quantitative in nature. So yes, we have the long short, and if you look at the long short in both the March and the November timeframe, there is a difference, but it doesn't appear to be all that dramatic. However, if you look below the surface at some of these types of radiomic features, you begin to see that there is in fact some relatively dramatic change. Volume has obviously significantly increased, density has significantly increased, and texture uniformity has dramatically changed. All of those are hallmarks of something far more progressive than might have been depicted, either in this qualitative read to the left, or even just looking at the long short with a more quantitative assessment. So looking below the surface, trying to understand what's going on at the tumor biology level, really getting clarity around that is the nature of what radiomic work is. When we're able to ascertain the patterns in those particular features, we can call that a biomarker. These are hallmarks to help us understand, again, the predictive, the prognostic, and the personalized opportunity within each of these particular analyses. So just to tell you a little bit more about how we do the work that we do, let me just show you a quick video. And so if you see here on the left-hand side, we've got 3 different time points. And those time points, you'll see the color map across the top. That's a texture analysis map that allows us to really begin to see the change in the heterogeneity and the texture of this particular lesion. But behind the scenes, what we're doing is the following. We're taking a look at the lesion, we're marking it, We're then doing a model to help us understand what's occurring within that particular lesion. And we're extracting data. This is that radiomic feature data I was referring to. And there are hundreds and hundreds of these metrics that are available to be used within the application or to be extracted, exported from the application for use in different analysis. On the right-hand side of the screen, you'll see a workflow that's associated with therapy response assessment. So very specifically, if you're trying to manage RECIST or PERSIST or any of those types of protocols, there are time point analysis that need to occur, and we're able to provide that information to you in a relatively automated way that allows you to really determine, again, very quickly, the differences that are occurring within those particular time points to depict whether or not you're actually seeing progression as you would expect or otherwise. So all of that is occurring within the application. And we're leveraging AI and deep learning to help us to inform the decision-making processes of our users. So first, we start with this patient imaging and region of interest analysis. So again, you're starting within the image itself. You're marking within the image what it is that you're interested in seeing. We're going through a segmentation and contouring process that accompanies that annotation and labeling process. We're rendering that in 3D so you can begin to see the structure and really understand it within the, as depicted within the anatomy. We're extracting the radiomic features. We're making those available for visualization so that you can see the various metrics as they are changing over time. And then we're also allowing for all of that data to be exported. So you can select from that 1,600 feature list that we provided, which of those that you want to export. And from there, you can then begin to do work outside of the system, or we can do that work for you as a service path. And at that point, what we're really beginning to do is bring together other data sets to help us understand the context of this radiomic information a little more fulsomely. And with that, we're marrying it with clinical data typically, or genomic data, different kinds of outcome data that is available either from third parties or from the organizations themselves. And we use that then to help us build outcome models that help us to try to present to you some particular areas where you can think about predictive or prognostic capacity. And so what does that mean? It means that we're going through a process of taking these 1,600 features, which are ascertained for every lesion or every tumor that we're taking a look at. And so obviously that's an awful lot of data. So the first thing we want to do is do a dimensional reduction to kind of remove some of the noise, as it were, those things that are not most important for you to pay attention to. Then we go through a process of selecting the model that we think is most appropriate to do the analysis here using kind of typical analytic capability. We then apply the information in combination with that model to give us a series of outputs. Those outputs are, first and foremost, a report. Here's kind of what we found and help you understand what's relevant to pay attention to within that data. We're able to get inputs for therapy response. So again, giving us some hallmark as to understanding whether or not something progressed as expected and quantifying it accordingly. And we can also trigger decision support alerts. So if there's something that we want to bring to your attention because we saw a rather dramatic change, we want to bring that either to the researcher or the clinician's attention, we can go ahead and do so. Here's an example of one of those outcome models that we did for glioblastoma work. So we were taking a look at 1-year survival and trying to understand which were the elements in the radiomic data pool that were most relevant and important to understanding that. And so we went through a process where we were able to ascertain those particulars, and it looked a little like this. We worked with the data from the Cancer Imaging Archive. We selected 90 patients. There were nearly 3,000 images associated with that. So if you think back to that cancer journey slide, it just highlights again how important the image is in terms of depicting what's occurred with those patients over the course of time. We then extract, again, 1,600 features from each and every one of those lesions as marked. From that, we distilled down to 195 relevant features, and that is then what we began our work in terms of the outcome model. We looked at patient survival, so again, the importance of bringing together different data sets to be able to take a look at that outcome information. And we discerned that there were really 6 radiomic features that were geometric in nature that really helped us understand what the prediction of 1-year patient survival would be. So these 6 radiomic features with a 90% precision, were able to help us understand that 1-year patient survival. So this is how you can take radiomic data and you can actually deliver new information beyond just that discrete data itself in terms of understanding what you can predict, what you can see in terms of outcomes, and how you might actually think about using this data to personalize treatment strategy. So there's a number of different use cases that we can share with you and how our clients are using it today. We work largely with life science companies as well as academic researchers. But we also have customers who are using the tool for early identification and screening purposes. And so let me walk you through a couple of these different kind of use cases. So as I mentioned, there's early identification and screening where we're able to help use the radiomics to help identify lung nodules, for example, in an early stage, and then put an associated workflow with that. We do treatment planning, whether that's using it for medical oncology or radiation oncology, as we'll talk about an example of that in just a moment. We can use our therapy response capabilities to help support disease progression monitoring. So if you're doing large-scale studies and you're dealing with criteria such as RECIST, DiRECIST, PERSIST, Lugano, and so forth, we have tools to help assess for that. We can use this information then to help inform clinical trial design, the arms of those particular trials, as well as any adaptive trial design that you may want to implement as a result of that information. And again, we've shared examples and we'll continue to talk a bit about predictive outcomes and how those information points can be used in drug and therapy development. So we're doing sort of, again, just to remind you, kind of 4 key processes. We're segmenting the image, we're extracting the radiomics, we're helping in the biomarker identification, and then translating those radiomics for use for our end users and stakeholders. This is an example of a study that was done at Moffitt that used our imaging segmentation capabilities to help them discern new radiomics that predict lung cancer survival in low-dose CT screening. And so they found 2, one intratumoral and one peritumoral radiomic feature that helped them really understand how to place people in low risk versus high risk groups in terms of 5-year survival. And so again, highly useful information as biomarkers to try to discern how people should be thought about in terms of their overall risk profile. Another example of a partner company that we work with, Reflection, in terms of how they do their treatment planning using radiomics. And so they're able to take this data now and to help them use that to plan how to deliver the radiotherapy in a more personalized and specific way based on the nuances of their particular delivery strategies. This is an example of a pharmaceutical company that's using our tools to help them curate their imaging pipeline so that they can create a data pool for radiomic feature extraction that can be used then in their drug development strategies and therapy response assessment strategies. We work with another company, Ancora Medical, that uses the radiomic data in combination with their own radiology expertise and oncology data to create new real-world evidence. And that information obviously is of interest to biopharma as well as other researchers. So we can be part of an ecosystem or we can be used independently by various stakeholders. But it's important to know that we believe that this is about bringing together this rich set of data in the context of these other important use cases and scenarios that can help complement the work of other companies or clinicians who are trying to crack the code, as it were, on managing oncology patients as effectively as possible. So as I mentioned at the outset, while we focus on oncology today, radiomics is highly applicable beyond oncology into different kinds of disease and condition areas and therapeutic areas of interest. And so we expect and will be expanding into these other areas as we progress our work with our current and future clients. So we're excited about the possibilities of radiomics. We think that there's just a tremendous richness here that can be leveraged to help again in that predictive, prognostic, and personalization work that each and all of us are very focused on as we try to help make the lives of patients better. So thank you so much for your time and attention today. If you'd like to learn more about HealthMyne, I would encourage you to come and visit us at www.healthmyne.com. So thanks again and appreciate your time and attention.